So later I will probably the process is I will introduce you first, and you can talk 45, 50, or 55 minutes, and any time that should be okay, and leaving about 5 to 10 minutes for questions. Or if it really depends on your convenience, it could leave 4 to 15 minutes or any longer. That is flexible. One more thing is we probably want to record your video, and later we may want to broadcast your talk to a wide audience by putting your recorded video on YouTube channel in our department. We just want to make sure you are okay with that. If you prefer not to make it public, we could also make it. We could also not public, make you talk public on YouTube video.
That's okay with me either way.
Okay. Then we will probably make it put your recorded lecture, recorded talk on the YouTube channel in our department. Okay, I think we can get started. So will still some students coming in, and actually today we are running a little bit overlap with our new department chair talk, which will end up after 10 minutes, I think. And so new faculty members and the new students will come in and slowly after in these 5 to 10 minutes. But I think we can get started. Anyway, the video will be recorded. So it's really our great pleasure to have Dr. Tao Ju to give us seminar talk today. And Dr. Ju is a professor in the Department of Computer Science and Engineering in Washington University in St. Louis. He obtained his bachelor and he obtained his bachelor degrees in science and art, two bachelor degrees from Tsinghua University in 2000, which is the best university in China, and his MS and PhD degrees in computer science from Rice University in 2005, sorry. And his main research is in computer graphics and with interactions with biomedical applications and agricultural applications. And he's particularly interested in geometric modeling and shape analysis. And he is currently associate editor-in-chief for IEEE transactions on visualization and computer graphics, and so as associate editors for many other top journals like Computer Graphics Forum, Computer Aided Design, Computer Graphics Models, and Computational Visual Media. And he also serves as program committees for many top conferences like CGraph, CGraph Asia, Eurographics. And his research has been mainly funded by NSF and NIH, and especially he has been a NSF career awardee. And we welcome Dr. Ju for his talk.
All right, thank you. Thank you, Guoyu, for the very nice introduction and for inviting me as well. This is indeed my pleasure and honor to share our recent work in the field of plant phenotyping with you guys. I have to start with a disclaimer that I myself am not a biologist, and I don't actually work in the area of image formation or imaging devices. I rely on my collaborators who are in those domains to work on these interdisciplinary areas. So my research, as Guoyu has already mentioned, is in computer graphics. In particular, I'm interested in creating and understanding 3D models. The reason I got drawn into the field of imaging, especially biomedical imaging, is because the process of getting knowledge from the images often involves creating 3D models and extracting features from 3D models. And in turn, the algorithm that developed there can serve back into the computer graphics community to solve other problems. So this has been really a rewarding experience for myself, working with my collaborators in biomedical image analysis. So I thought that I will start briefly by outlining my research agenda to give you a better idea of how this particular topic fits in. I know I don't have too much time, so I'll try to fly through things, and I apologize ahead of the time that I might be missing many interesting details, but I'll be happy to talk about them either after the talk or offline and in emails. So as I said, my research is in computer graphics, particular interest in creating 3D models. And this is generally the problem called with 3D reconstruction. I'm particularly interested in reconstructing from a simple and sparse inputs, because that means that you won't spend too much time or effort in creating inputs to get a nice and 3D models out. So I worked in areas of on problems like reconstructing 3D models from cross-sectional curves, which are typical output of manual segmentation in medical imaging, or from freeform curves in 3D created by virtual reality sketching tools. And more recently, we worked on creating piecewise smooth models, such as man-made shapes using sketching as well as sparse samples. Once you have the 3D models, lots of application domains require extraction of knowledge from the models, and often those knowledge comes in the form of a shape features, such as sharp features or semantically meaningful patch decomposition, or some form of compact representation of a 3D model, such as skeletons. And I also work on analyzing the other sort of discrete aspect of 3D models known as topology. So this deals with connect components, handles, and voids. So I worked on a few methods for cleaning up topological errors of 3D models, and something I'll be talking about today as well. The third bucket of my research is using the first two buckets and apply them to specific domain problems. I've worked on a few of these cross-disciplinary projects in the past. Some of the most sort of long-lasting ones include creating 3D structures of proteins from cryo-electron microscopy imaging. This heavily uses a skeletonization as an intermediate tool for creating and understanding where the alpha helix and beta sheets go into the volume given a three-dimensional imaging of the protein. And more recently, and of course the focus of this talk, is to understand the structure of roots of plants, again from 3D imaging. So this is where we are, and I'll be talking more about this particular project. And during this process, I'll also mention some other works I've done in the domain of shape and topology analysis. Now, these algorithms were designed to tackle the specific problem of root modeling, but in return, they can also be used for solving other problems in computer graphics as well. So hopefully I give you sort of a high-level view of how I get to where we're going to talk about today. And I just want to take one step up and bump up one level, because this work that I'll be presenting today falls under this bigger and rapidly changing the field called plant phenotyping. So you've all heard about genotyping, which is investigating the genetic makeups of plants and life forms. Phenotyping is a measurement of the visible aspect of plants, such as the forms and in some cases functions as well through other experimentation. The goal of phenotyping is to better understand how do plants grow and how do they interact with the environment. Essentially seeing what's the effect of the genetics, and correlating between genotypes and phenotypes is a fundamental way of pushing forward biology. Some applications are in agriculture, for example, how to breed the next generation of crops that are not only sort of resistant to drought, to pests, but also can benefit the environment. And how do we manage those crops, right? So these all relate to the forms and functions of the plants. The technology for phenotyping has really advanced over the past few decades, moving away from the lab setting, one plant at a time, to devices that can offer real-time phenotypes of plants on a very large scale, such as What you see here, we have robots, we have drones, we even have this large-scale field scanner that provides information such as 3D points or even hyperspectral images. So this effort has been going on for a while, but what's actually missing in all phenotyping and what's the underdeveloped part of plant phenotyping, in my opinion, is understanding the roots. So the roots are what's called the hidden half of the plants because they're hidden and they're as big as a plant. So that's a hidden half of the plants. Roots are very important for plants. They provide support, physical support. They also are the main channel for taking up water and nutrients for the plants. And in addition, they do a service for the environment. They stabilize the soil, prevent soil runoff. They enrich the soil with organic matters, and they are the primary way for carbon sequestration. Right? So there's a lot of good reasons why we should care about roots and why we should try to better understand roots and engineer more healthy and stronger roots. So when it comes to roots, the structure is very complex, of course, and rather unpredictable, unlike the above ground part of the plants, which everybody is familiar with. There are many aspects of the shape or the form of the roots that biologists do care about. So these traits, what we call morphological traits, are the aspects of the forms of the root. They typically fall into two different categories. There's one category of traits which care about the overall shape of the plant, such as depth, width, volume, or convex hull volume, etc., or some averaging or statistical measures of the plant, such as the total number of root tips, the average direction of the roots and total length of the roots. So these quantities already give biologists a lot of information about the roots, but oftentimes there is need to dig deeper to understand, for example, the geometry and shape of individual roots. So that requires tracing of individual roots from a large root system. But even more importantly, roots grow in a structured way. So roots, they either grow directly from the seed region, or they grow from existing roots. So there is a natural or inherent ordering among the roots as the roots grow. The biologists are very interested in this hierarchy ordering of how the roots grow, because that tells them how the roots grow to what they are now. So this developmental aspect is extremely important for biologists. So being able to annotate, identify individual roots and annotate with its type and their ordering in this hierarchy is sort of the next level of traits that biologists really want to gather from the roots. Beyond that, there are also traits that are specific to type of plants. For example, corn roots have these nodes where the roots are growing from the stem. So they don't just grow anywhere on the stem. They usually cluster around these discrete locations called nodes. And there are other roots that have nodules on the ground for fixing nitrogen and different type of root features. And so those are quite specific to different species. So as you can see, there is a lot we can learn, or a lot we want to learn from roots. So how do we get these traits? Well, the common way, which has been going on forever, and people are still doing that, is to do it by hand. This involves digging the roots from the ground, cleaning them up, and then just remove one root at a time from the root system. And for each removed root, you use a caliper to measure the length and use some other measures to measure curvature. Now, obviously, this takes a very long time to do even for one root. And it's quite subjective, depending on how you cut, how you measure, you can get quite different measures. And last but not least, it's destructive, right? You remove the roots, and then the root is destroyed. You cannot analyze it again. So a better approach, which most of the more and more labs are using this approach, is doing imaging and performing computational analysis of the images. Now, they will offer higher throughput process. Results are more quantitative, and they're not destructive, right? The same root can be imaged again, and again. And even better, there's a potential for observing the root growing in real time if you set up the experiment in the right way, for example, imaging the root in the soil. So this is where my work falls in, is to help plant scientists extract useful traits, information about roots from their images. And because this is about imaging, I just want to quickly give you some idea about root imaging and what kind of images do we get by imaging roots. Traditional imaging uses two-dimensional methods. For example, Rhizotron offers a cross-sectional view of the soil. You can also do flatbed imaging of roots excavated from the ground. These two-dimensional methods, however, cannot give you traits that relate to third dimension, for example, volume or angle and directions. And it's very hard to obtain a hierarchy of roots from these 2D imaging because roots will be hiding behind another roots or hiding behind the soil. So a much better way, or the best way to obtaining the full 3D information is to do 3D imaging. And there are a variety of modalities and ways you can image roots in 3D. If you have excavated roots, so roots dug up from the ground and cleaned, you can image them using multi-view scanning or using a CT device like the one shown on the right-hand side. And I just want to show you a quick video so you get an idea of how much details can be actually captured by the 3D imaging. In this case, it's a CT imaging. This is an image produced by one of my collaborators at Danforth Plant Science Center here in St. Louis. Now, as I mentioned earlier, 3D imaging has a potential to observe the growing of roots in real time. And indeed, there are ways and there are labs actually succeeding in doing that. For example, on the left, I show the roots growing in transparent gel, which to me is an amazing way of observing how roots grow because you make the unseen invisible, right? Because now you can see the roots growing in real time. On the right-hand side are results of imaging roots growing in actual soil or media that's similar to soil, right? So with these imaging modalities, you can actually capture multiple time points of the same root growing over time. So with the increasing amount of the 3D images, the natural next question is, how do we best utilize these images? How do we get as many traits as accurate as we can, as fast as we can from these images? So that's kind of where the problem comes to us, is developing algorithms for producing traits, particularly fine-grained traits. Now, we're also interested in coarse-grained traits, such as total volume and depth, but because the images have so much information in there, it's kind of a waste not to being able to dig deeper into the images and obtain these fine level traits, which are also very important for biologists. This is a hard problem, of course. Naturally, the images are not perfect. They have noise. The resolution are almost never sufficient to capture the finest roots. There's also low contrast issues, especially if the roots are growing in the soil in different media. There's also the inherent complexity of roots themselves. Like I said, roots are complex and unpredictable in the way that there are a lot of study about the structure of the plants above the ground. There are even mathematical ways to represent those structures using parametric L systems, for example, or procedural models. But they don't apply to the roots because the roots, they grow in very different ways. You cannot easily tell what plant it is just by looking at its root, for example. However, there are some commonalities among roots. One of them that I'm gonna talk about and utilize is an important topological prior or structure prior, in that most roots, vast majority of roots, they don't have loops. They're connected, but they don't have loops or cycles that kind of the two branches, they start from the stem and they merge into one. That usually never happens, right? So this is an important sort of prior and also constraint for modeling roots. And last but not least, for root phenotyping, you need to understand biology of roots and biology of specific species to incorporate the knowledge into the phenotyping pipeline. So with these many challenges, it is not surprising that the majority of the methods to date can only produce coarse-grained traits, such as total volume, depth, total number of roots, because these traits, they don't require an accurate reconstruction of the root in the first place. You can make some errors, you can miss some parts, and you can incorrectly merge some roots together, because the overall volume does not change much when you make small mistakes in the initial processing steps. However, if you want to dig into the fine level grade traits, you would need a much more careful processing stage to give you that fine level traits. And as far as we know, there are only 2 methods out there that can produce fine level traits, but some of them, like this dynamic roots, is only designed for very simple seedling stage root systems, usually grow in transparent gel in a small container. They don't grow very big, so analysis is usually a lot easier, and the system is a lot simpler as well. Or you have methods that can only produce the top level of the hierarchy, because the method that we're using are based on visible lights, and they cannot really see through the crowded crown into sort of the body of the root system. So there are no methods that we know about that can produce a complete branching hierarchy, as well as the other fine-grained traits directly from complex roots, such as those actually growing in the field. And this is where our work come in and to try to fill this gap, or make a step towards filling this gap. Is we created this tool called a topo root, and I'll explain why it's called topo root in a minute. But it's a software pipeline that takes 3D CT images of mature root systems of maize or corn, such as those excavated from the field, and will produce a complete set of hierarchy with different root types and associated geometry measures for individual roots. So the current system is designed for CT images and for corn roots, but we definitely think it will be possible to extend the system to other type of species, plants, and other type of imaging modalities as well. All right, so going into the pipeline, it consists of 3 major steps. The first step is taking the input 3D image and produce a reconstructed 3D model of the root. The emphasis in this step is to try to create a model that best matches the topology of the root. So like I said, the root is a connected shape that doesn't have handles, that doesn't have loops or cycles, right? So we try to make sure that we achieve that particular structure and topology as best as we can at the reconstruction stage. In the second stage, we'll convert the 3D model, which is usually represented by voxels or by triangular meshes. They're not that easy to analyze, so we're going to turn the model into a what we call a skeleton. A skeleton consists of 3D curves that lie in the center of the individual roots, and the curves are connected in a way that they preserve the connectivity of the root itself. With the skeleton, we can now analyze things such as tracing individual roots, building hierarchy, and analyzing geometry of individual roots. So skeleton is an intermediate structure or shape representation that enables and everything that we do later on to compute different traits of the root. I'm going to focus more on the first 2 steps here, because the first 2 steps, which is reconstruction and computing the skeleton, they involve development of new algorithms in computer graphics that I have found to be quite useful even for solving other type of problems outside plant phenotyping. The last step is not as complicated, but I will just briefly touch upon it. So in the rest of the talk, hopefully I will spend maybe 15 to 20 minutes. I will focus on the first two steps. All right. And by the way, I don't know how this symposium or seminar is usually run, but if you have a question, I will be happy to answer that question in the middle of the talk, or you can type in the chat box. I'll be happy to, if I get a chance, I will look at it and try to answer it. All right, so the first step is reconstruction. Now, there are lots of image segmentation algorithms out there whose jobs are to separate the foreground from the background in possibly noisy and low resolution images. Now, those segmentations methods work great, except that they don't have much guarantee of the quality of the segmentation, especially in terms of topology. So when I talk about topology, I'm referring to connectivity features such as isolated components and cycles, or what we call handles, and internal voids or cavities. The reason we care about these topological features is that in order to analyze the branching hierarchy of the roots, it's very important that you start with reconstruction that's as close to a root topology as possible, meaning that the reconstruction should be connected and should not have any loops or cavities. However, typically, segmentation methods would give you results that contain lots of these topological features. Even for CT images of cleaned up excavated roots like this one, where you can easily get the nice segmentation by thresholding, you can still get topological errors like handles, right? Where two roots that are very close by, they appear to be touching or merged in the image. In reality, they don't, but because of the noise and the resolution of the imaging, they appear to be touching, so that forms a loop. Or you can have thin roots that appear to be separated and disconnected in the images, right? Of course, again, in reality, they don't happen, they're connected. But again, because of imaging, you have these islands that we call, which are disconnected components. And even though there only appear to be a few of them in the image, by quantitatively, you can measure the number of these features, and there are a lot of them, thousands of them actually, spreading around in one root images. So of course, having these many islands, handles, and cavities will not help us in getting a good hierarchy structure out of the root. So the problem is how to remove these, what we call topological errors or topological features that shouldn't be there. And the goal is to maximally remove them without destroying the shape of the root. You still want the reconstruction to be as close to the original image as possible, but you want to remove those components, cavities, and handles. So there are two goals at hand. One is topological and one is geometric. So how do we remove a topological feature? There are actually two ways of removing a topological feature. One is to adding stuff into the object, the other, which I call filling. The other one is a complementary action, which is taking away stuff from the object, which I call cutting. So I'm going to show you a very simple toy example to explain what is cutting and what is filling, and that will help you understand how topological simplification is operated. So let's say we want to remove an island. There are two islands here. Let's say we want to remove one. Well, the cutting approach will be just completely remove one of the islands, take it out, okay? And then you reduce the number of islands by one. The filling approach will be building a bridge that connects between two islands. You will also reduce the total number of islands by one. So that's cutting and filling. For the handle, you can cut open the body of the handle, or you can fill in the kind of the donut hole in the middle of the handle. Both would reduce the number of handles by one. And similarly for cavities, you can either dig a tunnel to reach to the interior cavity that would connect the cavity to the outside and thereby reducing the number of cavities by one, or you can just remove the cavity, fill up the entire cavity, and you can also, that will resolve that topologic feature as well. So these two ways are both possible. The choice of which one to use would depend on which way would minimize the change of the geometry. Right? And this is where different methods differ. So looking at the previous works in this area, that there are a lot of methods actually working on simplifying topology of 3D models. A group of methods only perform cutting or filling. So they either add stuff to the object everywhere, or they remove things from the object everywhere on the object. They cannot do adding in certain places and filling in other places. So these are what I call the monotonic methods. The downside is that they can cause substantial changes in geometry. So take this input as an example. This is a blood vessel from MRI angiogram. And you can see there are topological features such as islands and handles. There are many of them actually. Now, if you only apply filling, that you're only adding stuff to the object, you can end up creating a large fill just to get rid of a handle. So think about that handle in the middle as a huge donut with a very large donut hole, because you can only add things to the object to remove handles. Your only choice is add a large fill in there. And also, you end up keeping all the little tiny islands on the outside, which might be actually imaging noise, but you're forced to include them in your final reconstruction because you cannot remove things. On the other hand, if you only do cutting, the noise are gone, right? You don't have to add a large fill in the middle, but you also remove any island, including those that are really big, which should be part of the object, and like those in the red boxes, so they're gone. The second group of methods are what's called the non-monotonic methods. They can choose whether to perform cutting or filling at different places of the object in order to minimize the geometric change to the shape. So some of the methods, in fact, existing non-monotonic methods, they use all kinds of greedy heuristics and try to reduce topology without changing too much of geometry. And they can improve upon existing monotonic methods quite a bit, as you see in the green boxes, right? They're, to get rid of that handle, they made a small cut on the vessel. And to resolve the island at the bottom, they build a bridge. So they're already choosing between cutting and fitting in these two different places. But the solutions are often suboptimal. For example, in the top red box, we're still missing a major component of the vessel. We developed a new method, well, not to solve the MRI angiogram problem, but for solving the topological simplification problem in general. And in this algorithm, we formulate a global optimization problem in terms of, given a set of cuts and candidate cuts and fills, what are the optimal ones to choose such that you can maximally drive down the number of topological features while minimizing the total geometric cost. We're able to formulate that as a graph optimization problem, and we can solve the problem by converting it to another well-known graph optimization problem called the node-weighted Steiner tree, and thereby getting much less suboptimal results than existing greedy heuristics, as you can see in here. I'm not going to go deeper into the algorithm, but if you're interested, you can either read the paper or I'd be happy to talk about it later on. But just showing some examples of this particular method applied to the roots. So this is the root scan that I was showing you early on. And then this is on the right are the results of applying this particular simplification method using global optimization. As you can see, the handle between the two nearby roots are removed by separating out. So I'm going to remove the line. You can see there is a separation now along this line between the two roots, so that handle is resolved. On the other hand, the small islands are connected up to form a contiguous thin root. So this method is actually very successful in reducing the vast majority of topological features in the input. So now we have removed thousands of those features, left only with 3 handles and 32 islands. So of course, the method is not globally optimal yet, because the problem that we're trying to solve turns out to be an NP-hard problem. So there is no hope of getting the optimal solution every time. But that's an area of area that we wish to improve in the future, is to devise more and more optimal algorithms that can solve that global optimization problem. The other limitation of this method is that sometimes it makes a suboptimal choice. So if you look at the dotted box on the right-hand side, our algorithm decides to cut a root branch in the middle in order to remove one of the handles. And of course, that's not a reasonable thing to do, but our optimization algorithm is guided towards minimizing the number of voxels that are changed between the input and the output. And for thin roots, it's very easy to make a wrong call by breaking the root in the middle in favor of protecting somewhere else that should be broken, but that would involve more voxels. So another area of improvement would be to try to find out better ways of assessing whether a cut or fill should be used or not. All right, so coming back to the pipeline, now we have a reconstructed root model. The next step is to represent using a graph called a root skeleton. Again, this process, this particular step is necessary so that we can perform analysis such as tracing a roots or identifying a branching structure, all of which needs to understand where do roots meet and how they're connected. It's much easier to do that once we have a graph representation of the root, such as a skeleton. So the skeletons are quite common shape descriptors in computer graphics, in computer vision, and in medical imaging. In the specific case of roots, we would like the skeleton to have the following properties. The first is that the skeleton should capture well the geometry of the individual roots, because we're going to analyze the roots using the skeleton, such as measuring the curvature or tortuosity of a root. So we need a skeleton curve to be able to capture those properties. And more importantly, in order to analyze hierarchy and branching, we want the topology of the root in the reconstructed root model to be exactly preserved by this network of curves. We don't want this network of curves to create additional components or additional cycles in the graph so that we can accurately sort of analyze its hierarchy. And last but not least, there's a practical concern because these root volumes are huge. So there are at least about 1,000 voxels in each dimension. So we really want to have efficient algorithms that can compute high quality skeletons from very large image volumes. All right. Okay, so there was a question by Robert. The image modality was used in example is X-ray CT imaging. All right, I hope I answered this question. I'll be happy to come back to it later. Okay, so there are many skeletonization algorithms out there that one can use. What we found out is that it's not easy to find one that can, number one, preserve the topology of the object, and number two, be very scalable to large inputs, such as a root volume. And I will be more specific about that. Overall, the skeletonization methods can be grouped into two different classes. The first class are methods that work on object represented as a set of voxels. So those are what I call voxel-based methods. And usually they work by erosion or thinning process, by which layers of the voxels will be stripped away from the object until you're left with a set of core voxels that either form sort of curvilinear structure or sheet-like structure, like the ones I show here. These methods are quite simple to implement, and they can exactly preserve the topology of the voxels that you start with. The downside, however, is that because it has to process all the voxels interior to the object and store information on it, it can be very time-consuming. And the complexity of these algorithms usually scale cubically with the increase of imaging dimensions, because that's a growth of the number of voxels as you increase the dimension. So this approach works, it seems to be perfectly suited for our purpose because our input are images and the voxels. They cannot handle the size of volumes that we're operating at. The second class of methods use only information on the boundary of the shape. And these methods work using a concept called Voronoi diagram, which is quite simple, and I'm going to explain here. So given samples in 2D or in 3D, the Voronoi diagram consists of all the locations or points that are closest to two or more samples, right? So those are the sort of in the middle of those sample points. And methods relying on Voronoi diagrams typically are variants of the following scheme. Is you place samples along the boundary of the shape, those are the black dots there, and then you compute the Voronoi diagram of those samples and then take a subset of those Voronoi diagram. It works really well in 2D, And it's very efficient because the complexity of the algorithm scales only with a number of points on the boundary of the shape, not on the interior. So the complexity of these methods usually scales quadratically rather than cubically with the increase of the size of the input. However, this method often fails to capture the topology of the object, especially in 3D. There are some inherent challenges of extending this 2D approach to 3D, which I will not detail, but I'm happy to talk about later on. And just to give you an example, these two methods that I show down there are two of the most popular or successful methods for computing a skeleton of 3D object using this Voronoi-based approach. And by the way, the skeletons, they look like sheets, they don't look like curves, because that's what they are. This is a specific type of two-dimensional skeletons called a medial axis. Now, the numbers on top of models show what's called Euler number, and that's the number of islands plus the number of cavities minus the number of handles. So as you can see, the original model has -3 because it has the 4 handles and 1 component, but none of these methods can reproduce that number. In fact, they either produce a lot of disconnect components, like in the method in the middle by Day and Zhao, or they produce lots of voids, like the method by Amanda and Kaluri. So in summary, there's just a gap there for methods that can first preserve the topology of the model and second, be scalable to very large inputs. And this is again, where we design methods to try to fill in the gap. So we design a method that takes 2 steps to achieve this goal, take a 3D model, and we output a skeleton that preserves topology and be efficient at it. The first step is taking a 3D model. We first compute this medial axis, which I just talked about. It's a 2-dimensional type of skeleton, but our method is like one of those boundary-based methods using Voronoi diagrams. The difference is that we can actually give a theoretical and practical guarantee of topological preservation. In this way, we don't lose the topology of the object, but we can be as efficient as these boundary-based methods. In the second step, we take the 2-dimensional skeleton and reduce it down to a bunch of curves and surfaces. The amount of curves and surfaces can be controlled by the user. This process, again, guarantees to preserve the topology, which makes the entire pipeline topology preserving and being efficient. I'm not going to, again, into details of either of these methods, but I will show some examples, particularly when applied to the roots example. So on the left here, you see a CT scan of the cone root. This is segmented CT scan using a simple the thresholding technique. And on the right, you see the medial axis computed using our first step. Remember, the first step is taking 3D model and reduces down to 2-dimensional skeleton structure. And it's very hard to see at this kind of scale. So I'll do a zoom up and you can kind of see the resolution of the voxels on the input and the details of the medial axis on the output. Now, this is all done quite efficiently, taking less than a minute. And as I mentioned early on, existing voxel-based methods based on thinning and stripping voxels, they have inherent challenges going to working on large volumes. In fact, none of the existing voxel-based methods can handle volumes this big. So the fact that we can handle this size of data in under a minute is really important for putting together the pipeline for root phenotyping. And combined with the second step of reducing this 2-dimensional skeleton down to 1-dimensional curves, as you see on the right-hand side, we complete the process of going from 3D models to 1-dimensional curves. On the far right here, the coloring of the skeleton is by the thickness. So thickness is measured as the shortest distance from every point on the skeleton to the surface of the original input. And this is very useful for analyzing the traits of the roots, as we're going to see next. All right, so now we're done with the first 2 steps. I think I kind of met my goal of in terms of timing. And then I'll just briefly touch upon the very last step of taking this graph representation with attributes such as thickness and obtaining fine-grained traits of the roots. This is done broadly in 2 steps. The first step is identifying where is the stem region. So we're focusing on corn roots here. So corn roots have a very thick stem, so that's quite easy to pull it out from the skeleton. As you can see, the red region on the skeleton is essentially where the stem is. We extend that stem to a stem region to include all the skeleton edges that are kind of located in the body of the stem, because they are basically, representing the stem itself. Now, the more interesting question is, how do you identify the hierarchy of the remaining roots? We treat the stem as level 0 roots, right? Because that's kind of where the other roots are stemming out from the stem. And the problem of computing the hierarchy boils down to the following question: is at every junction of the skeleton. So I'm going to assume that at this point, the skeleton is already simply connected without any cycles, right? So it's a tree, basically. And we're going to treat the stem region as level 0. And by that, we can compute a direction on every edge of the skeleton that's coming from the stem, right? So it's becoming a directed tree. So at every junction of this directed tree, we have to make a decision regarding which one of the outgoing edges is a continuation of the root represented by the incoming edge. There is exactly one of these outgoing edges that does that, because the way that roots grow, as I said early on, is growing from an existing branch, right? So at any junction, there's going to be an incoming edge, an outgoing edge that forms this branch that was grown first, right? And the other outgoing edges are growing later than this first branch. In terms of the level, that particular outgoing branch who is continuing the incoming branch will given the same level as the incoming edge, and the other outgoing edges will given a level higher, right? As shown in this diagram. Now, this is a very ill-posed problem. We don't have much information about which outgoing edge we pick. This is where we consult with biologists and ask, what do you think? Right? What are the rules that should govern the hierarchy? And they told us two things. One is that usually in a root, the maximum level of any root is not large, right? So we want to minimize the depth of the hierarchy. This is sort of a topological prior. And the second is that roots at lower levels are usually longer because they have more time to grow, right? So that's a kind of geometric prior. And combining these two prior, we came up with a discrete optimization algorithm that infers this choice at each junction and come up with the hierarchy. So in the picture on the top right here, the coloring indicates hierarchy. It's a heat color. So the more blue it is, and that means that the level is lower, and more red it is, the level is higher. Okay, so that completes the pipeline. I'll briefly touch upon evaluation. I know I'm running a bit short on time here, so I might skip one or two evaluations. So the software pipeline was evaluated on an actual corn roots excavated from the field here in Missouri, actually Illinois. I think they crossed the state boundary to that's where their fields are. And here are some examples of the roots. And the coloring again shows the hierarchy that are computed from our software. They actually planted two different types of corn. There is a wild type, which is a typical corn, and there is another type where they did a genetic modification so that these roots tend to produce fewer nodal roots. So nodal roots are the roots that stem directly from the stem, or what we call the level 1 roots, right? And part of the exercise is to see whether a computational tool can distinguish these two types of roots, have a way to count the number of nodal roots and show a sufficient difference between that count, right? So I'm going to first compare sort of visually the result of our method with some other methods, such as dynamic roots. As I mentioned early on, dynamic roots is another method that can produce a full hierarchy, but it's designed for seedling roots, simple systems. So it has a lot of trouble on complex root systems like the one we're shown here. These are mature grown corn roots. So as you can see, not only it misses a huge part of the root down there, and this is because they operate directly on input reconstruction without doing any topological repair, but they also mislabeled a number of branches. These branches should be at least level 1 or level 2, but they've been labeled as level 0, just like the stem. We did an additional experiment by feeding in dynamic roots with our topologically simplified reconstruction. So we're trying to take that error away from dynamic roots and see how it behaves. And in this case, because now it has a more complete shape, it doesn't, it does not miss a major part of the root as it did before, but it still makes quite a bit of mistakes in this area as well as in the area above it in terms of labeling the roots by its hierarchy level. As a more quantitative way of measuring, we actually asked some graduate students in Danforth Plant Science Center to manually measure the number of nodal roots of those corns using the process I talked about early on, right? They have to use a pincer to cut away the branches and manually count them. That took several days. So just an example of the difference between manual counting and image-based analysis. And we plot this correlation graph where every dot on the graph represents one corn root system, and the x-axis is a manual count, the y-axis is a count produced by our tool. And as you can see, it's a very high correlation between the two sets. And it's very clear now that the wild type has more nodal roots than the mutant. And that can be done seen either by the manual count or by the results produced by the topo root system. And in contrast, the dynamic roots, even with topological simplification, fails to show any level of correlation with the manual count because the errors it made in labeling the hierarchy levels. So this is quite encouraging. I don't think I'll have time to go through the remaining set of experiments, but I'm just going to kind of go through, briefly touch upon some future works, and I'll conclude the talk and leave some time, a few minutes for questions. There are a lot of open questions in this field in terms of root phenotyping, root image analysis. One is working with challenging imaging data. For example, the picture you saw on the right is a cross-section of a soil core with roots growing in there. So there are actually roots in there. Unlike CT imaging of excavated roots, segmented roots in soil is extremely challenging. For example, I would challenge you to find out where the roots are in this picture. I couldn't when I first saw this, but now I'm going to turn the roots on and you will see if your guesses are correct. So you see those blue dots, those are the roots, right? I'm going to flip back and forth, right? So that's how tiny the roots are. So segmenting those roots out is a very challenging task. Other challenging examples, including multi-view imaging of this fibrous root system. Lots of thin roots, just like hair strands, hanging in the air, and you have to scan them. To produce good root models from these challenging examples, these new algorithms, perhaps deep learning can help, and perhaps new algorithms for computing skeletons and repairing topology are needed as well. The second direction, very exciting direction, is time series, right? So if we can observe roots grow in time, can we do this type of fine-grained phenotyping for each time point, but making sure there's temporal coherence, right? So, this require new algorithms for repairing topology, computing skeletons and hierarchy that grows in time and does not grow backwards or suddenly kind of flip back and forth. So again, there are very interesting challenges for algorithm development here. And last but not least, how about above the ground parts of the plants? We can also use these techniques we developed or new techniques to do fine-grained phenotyping for the above ground of the plants. This is an example of a publication that we had early on for the sorghum panicles, showing the little insert here. And we were able to use a variant of our technique to analyze the primary branches of the sorghum panicles. So that's just an example of techniques for underground can be transferred in some cases to parts of the plants above the ground. That's it. I would thank all my students for their hard work. I did not code anything, so it's all their work hard work putting there to make this happen. I thank my collaborators. I want to give a shout out to Chris Top. He's my main collaborator from Danforth Plant Science Center, which is one of the largest plant research institutes in the nation. And there is some amazing facilities there for root analysis and for imaging science. have collaborated from St. Louis University on the math side. And last but not least, I want to thank the NSF and various other units for their generous support of the work. And with that, thank you all for listening. And I hope I still have some minutes for answering questions. Thank you.
Thank you very much, Dr. G. Are there any questions from audience? Probably I get started. There is one. Tosi, you can go ahead.
So I had a question regarding the segmenting the roots out portion. So if my knowledge serves me right, the roots are like a meshed network of overlapping where there's a lot of overlap. So when you're segmenting, how I may have missed it, you may have mentioned it, but how are you considering the overlapping of the root networks?
Let's see. I'm trying to go to the slide. Okay, so the images that we use are 3D images. So I'm going to replay this video. This is kind of the input our method. So we don't have problems like occlusion as in 2D imaging. In 2D, you do have branches covering another part of a branch, and you have a problem with occlusion, but we don't really have that occlusion problem. We have a different kinds of problem, which is the imaging resolution may not be enough to capture the gap between two neighboring roots. So we can have places where the two roots appear to be touching or merging, even though they don't overlap because they don't grow into each other. Mm-hmm. So that's where we have to worry about topology and removing those topological errors. Does that answer your question?
Yeah. Another follow-up question to that would be like when the roots get extremely thin. I recall in one of your slides, you showed like there was like a part of the root that was broken and it registered as like two different bits. So how are you like, so from a human's point of view, we know that was like a root that was missed by the algorithm, but how does the algorithm like adjust to that basically?
Yes, so of course, I mean, this is one of the good examples, and I only show good examples in my talk, right? But in this case, if you notice that the distance between those islands is much smaller than the size of the islands themselves, right? And our optimization energy would allow our algorithm to pick the easier way of fixing a problem. Easier meaning that it involves a change of at least the number of boxes, right? So that's why it works in this case. We do not know, the algorithm does not know that they should be a branch. They just count the number of boxes between them and, you know, using that as an energy. So like I said, you know, this relates to one of the limitations of the algorithm is that sometimes it doesn't do that. Sometimes it can actually cut an intact branch into half just to minimize the number of topological features. This just, you know, there's a great example of you know, the fact that the algorithm is not smart enough to figure out that's actually a branch. So one of the follow-up works, and we're considering that, is how to build more intelligent ways of assessing whether a cut or a fill should be used. And that should concern the shape of the object, not just the topology.
All right, yeah, that answers my question. Thank you.
All right, thank you.
Any other questions? So actually, I have a question. So when you talk about the 3D reconstruction part, you talk about using the topology to remove islands and handles, and before that, and from the CT imaging and to the 3D model, and I want to know whether there's some specific magic things that you make the 3D models before doing those kind of cutting and filling.
Okay, let me get to the right part of it. Yes, so for this pipeline, we're dealing with the CT images of clean, excavated and cleaned roots. So we're assuming that the contrast of the images are high enough such that you can use simple thresholding to create a 3D model. Now, this is not the case if you look at other image modalities, like the one I shown at the end, right, like these ones, right? For example, you can't just use thresholding and hoping that you can grab the roots out. So how to incorporate this topological prior that the root is simply connected into a segmentation framework, whether it's a classical segmentation using region growing or the more modern versions of segmentation using deep learning, that's a really interesting direction to go into.
Also, I have another question. And using the topology, I see there is a very good reason to use the topology from the geometric perspective. And from plant science perspective, though, will that topology still have some specific topology from the plant science perspective? For example, the root, whether there's some specific topology that should exist in the root of one root type, one species, and versus another species, whether there should be some different topologies.
Yeah, it may not just be topology, it could be the structure, right? For example, if you look at corn roots, they look very different from rice roots, for example, because they belong to different classes of roots. And there are certain classifications of root types depending on the plants. And those are very useful information to incorporate into the algorithm. We haven't gone to that stage yet. Right now, we're just starting almost from nothing, as if you know nothing about the roots, except that they are simply connected and without handles. And the next logical step is kind of tailor the algorithm to a specific category of roots where you can utilize those prior information in certain ways. Although there are problems overfitting, right? Because you kind of use too much, then you always produce roots that look like kind of the template that you start from. And that's not good for experiments where you have, you know, genetic mutants versus wild types, and you really want to look at their differences. So I would say that there are a lot of information, but we have to use those information wisely.
Yeah. Another questions from the audience? So if there is no further question, let's thank to Dr. Ju again. And Dr. Ju will stay here for a little bit, several minutes longer if someone wants to chat with Dr. Oh, there's Nathan, you have a question actually.
Oh, no, I was just applauding.
Oh, yeah, sorry.
Thank you.
Then if any of you guys want to stay here a little bit longer to chat with Dr. Q for any questions, and feel free to stay here a little bit longer. Otherwise, that is all. Thank you, Dr. Q, for your talk.
Thank you. Yes, I'll be here for a little while.
I guess I do have one question.
Yeah, go ahead.
So I know you used excavated roots for this imaging. Is that considered technically destructive sensing, or is it somewhere in between?
Right. So there are different levels of destruction, right? The level of destruction I was talking about earlier was you're actually, you know, destroying the entire root by removing one branch at a time. Right? And then you're left with just the stump of the root, and you cannot recover the original shape of the root anymore. Now, what we do here, we're just excavating it. And because we cannot take the entire root out, root is usually huge, they took what's called the root crown, right? So that's a kind of in part of the root that's immediately surrounding the seed region. That's a part that they can feasibly be dug out of the ground. And they clean it, but they don't cut the roots away. They just keep the roots as they are. They dried it, they put it in the bag, and they send it to the imaging facility. And those roots can be imaged again, using different modalities, and even be dissected later on if you want to. So in that sense, it's preserving the root. It's not destroying it. But there is another level of destruction in the sense that the roots are dead. They cannot regrow again. You cannot put it in the ground and watch it grow and dig it up again to look at another time point. So this excavation is destructive in a sense that it kills a plant. Even though it preserves the root shape, it kills a plant. The time series imaging methods, like I shown on one of the slides, where you either grow the roots in the gel or you grow the roots in the soil and the image over time, those are the truly non-destructive methods, which are the direction that a lot of the root scientists are going after these days, because you can actually see the development of roots. That make sense?
Yes, thank you very much.
Sure.
So if there's no further questions, I just stop the recording.
Okay.
Actually, one of your questions, one of your points for future things is very interesting from the temporal things. Actually, temporal things is really, it has to be non-destructive. Otherwise, that it is very difficult to get to really get the temporal information.
Yeah, yeah.
You take that out and that totally destroyed, and you cannot.
Right, right. And the current this in vivo or in situ methods of imaging are limited to very small roots. Because if you want to monitor the roots growing over time, you have to keep it in container, right? And those containers usually are fairly small in size. So you cannot grow a large and mature plant. There were some images I didn't show where you can see that sometimes the roots are a few times larger than the plants themselves, like a grass roots. They are huge. So it's impossible to constrain them in a container. I think the next breakthrough should be some method of imaging, in vivo imaging in the field, in the sense that you don't have to put the plant in a container. And maybe that, that's where you're heading, Guoyu, is using GPR, the ground penetrating radar, right, to actually see them in the field without having to dig them out and not limited to how big they are. But even my point is the current method for these time series imaging is still quite limited to the size of the plant.
Actually, I see Yang is here. I actually want to introduce you to Yang, which is one of our really major faculties. Yang, are you here still? I saw he is here, but I'm not quite sure whether he's.
I have a question here by Robert. I don't know Robert's still here, but I don't know. So
he's still here. He's still here.
Okay, I can answer that first, and maybe we can wait for Yang to come back. And so Robert, you're asking about point cloud of roots and generate a hollow mesh, but you want to create a skeleton using from the point clouds, right? Is that your question? Is how to create a skeleton from point clouds? Okay, he's not. I guess he's muted, but the answer is there are methods. There are many methods for creating skeletons. The one I presented today are sort of the major buckets, but there are methods for creating skeletons from incomplete point clouds, which I can give you pointer to. Although those methods, again, are quite sensitive to the quality of the point cloud in terms of the sampling density, the noise level, and whether it has missing pieces. So there is usually no guarantee whatsoever about the quality of the skeleton you get from those algorithms, but it's worth a try. They don't need an actual complete mesh or a solid shape to start with.
Yeah, actually, that relates to one of my questions. Actually, yeah, I also want my student to try to use the 3D skeleton to 3D models to get the skeletons, but my students always tell me there's some problems in the currently existing algorithm. She tried several ones. She still have some problem.
Yeah. It's still an active research problem in computer graphics. So.
Yeah, Fei, you can go ahead. It's a chat. Don't worry too much.
Thank you, Dr. Zhu, and thank you for your wonderful presentation. And I got many inspiration from your slides. And I noticed you also had some picture about the drone images. I don't know if your project ever deal with any data from drones, because like Robert Chancia is, we are in the same group, and we are both under Dr. Yan Bernard. And our data are mainly from UAS-based point cloud, like that. I don't know, like in your past experience, if you have any algorithms or methods, dealing for the drone-based point cloud like that.
Point clouds, you mean just like the picture I show on the last slide, and there was this point clouds of
yeah, because I know like your input is CT images, right?
Right.
And
right.
So when we collect our data, we are mainly collect the ortho images, multispectral images, or LiDAR point clouds like that. And so in that case, we have many occlusion, and also our data may not be that accurate, you know, it's
Right.
because we are collecting it from far away, so like that.
Ah, okay.
Yeah.
Yeah. I can give you pointers to some algorithms. Like I said, there is no guarantee that they will work. And another issue with these, you know, there are now quite a few algorithms that take point scans and produce skeletons. They usually come with just some parameters, and you need to be able to tune those parameters carefully for your own data. And that's something that will take some time to get it to work. There are also deep learning algorithms now that take point clouds and produce skeletons as well. But then again, I think there is a question of training data and what you train with. So I don't have much knowledge in that area. So it's, you know, I can give you some pointers though.
Yeah, thank you. Thank
Yeah.
you. And I don't know, like for the deep learning method, it seems like collecting the ground data is a great, you know, a huge workload.
Yes, just annotation and, you know, producing ground truth is major issue. Although for, you know, if we're talking specifically about computing skeletons or point clouds, you can automatically generate training data with ground truth, right? By taking existing 3D models, right, and subsampling and adding noise to it. And there you have the ground truth skeletons. So.
Got it. Okay. If you don't mind, may I have one more question? Like,
yeah, go ahead.
Like, I don't know, like the project you just presented seems so cool, but I don't know, like, how about its connection to the industry application? Like, I was wondering, like, how far from your work to the industrial application, like?
Well, right now we're focusing on sort of doing fundamental plant science, try to understand the differences and features of certain, you know, plant roots. So we are not in the stage there that can be deployed by, let's say, an agricultural ag company, digital ag company to, you know, do this on a daily basis to screen their plants. You know, it would take quite a bit to understand their needs and the specific features they want from their plant roots, and that I don't think in general plant root phenotyping has been industrialized. It is still very much a research lab a dominated field. Unlike, you know, the typical sort of the high throughput field phenotyping, like above the ground phenotyping, there are already companies making robots, drones, like I showed in the picture. For plant roots, I think we're not there yet. That's why we try to push it so that we can catch up someday.
Thank you. Thank you.
Sure.
The algorithm can be used for many other applications. Actually, I believe there's much broader impact for the algorithm you have already introduced that could be applied to plant science, probably for later for robotics, for many fields, I think.
Yeah. I mean, that's why I love this type of work is you know, you derive you know, computational problems from a, you know, biological problem, right? And then the solutions to that could be generalizable to other areas. So that's probably my #1 sort of strategy of finding new problems for my computer graphics research is, you know, work with biologists and doctors and see what problem they have.
Exactly.
Yeah.
Let's see. Yang, are you here? So I couldn't see Yang. Actually, Yang is one of, I would say, in our department is the most agriculture related faculty member, and he and me has worked on agriculture things, and he's working on right now LiDAR and remote sensing, but his background is forestry right now. It's actually from forestry, and he has worked on that. That is why I really want you to introduce you here, but it seems like Yang is somehow maybe because today is a special day that our new director is visiting right now. It's something like interviewing our new department chair.
Okay. Well,
I don't know whether that changed make some changes. So, but
Yeah. No, that's fine. I mean, maybe you can connect me with Yang later.
yeah. Probably later I can send an email and to connect with you and Yang and see whether there's some collaborations in the future we can do. Yeah.
Yeah, that sounds good.
Cool. So anyone has some specific any kind of discussion? Actually, Dr. G is also currently serving as department chair, associate department chair right now, and used to be associate dean, right?
Yeah.
Okay, probably if no further questions, and we can end up here, and later I can send emails to connect with some other faculties and who probably has some collaborations with you.
Appreciate it. Yes, thank you. Thanks again for inviting me. This is great.
Thank you for a great talk. It's really somehow, we don't have many in our department. We don't have many. Actually, we don't really have undergraduate students. We don't really have much students. Maybe every year there's 1 or 2 master students, and then most of them are PhD students. And right now, I was tracking the attendance is somehow around 30 students, 30 people coming here. So it's it looks like in computer science that is small, but it's in our department is already a good.
You did a great job in advertising this event.
Well, I think that is because the topic is very interesting and people really want to hear that.
Yeah, great. Well, thanks again.
Yeah, thank you.
All right.
Thank you so much, Dr. Zhu
Bye.
and Dr. Lu.
Yeah, bye.
Bye-bye.
Bye.